Much has happened in this disease, and I think it really reflects the tremendous success of whole genome sequencing that allowed us to understand the genomic landscape. For the longest time, we'd have people asking us, well, what causes Waldenström's? And we had absolutely no idea. So at that time, most of our therapeutics had to be borrowed. I call it the hand-me-down treatment approach. And we would wait for our colleagues to vet out drugs in the myeloma field, in the lymphoma field, CLL field, in fact, any field. And as long as we had some credible rationale, we would then begin our own clinical trials. But this clearly wasn't what was making us, giving us the ability to move the field ahead, and understanding the genomic landscape was very critical. So whole genome sequencing was made possible because of the philanthropy of a donor and the International Waldenström's Macroglobulinemia Foundation, giving us at the time the money when whole genome sequencing was still a very nascent art. And this was a very pivotal moment in the history of Waldenström's because it allowed us to be able to identify some very key genomic mutations, the most important of which is a mutation in the MYD88 gene. This is a mutation that's found in about 95% of all Waldenström's patients. And it's a very important mutation because it turns on important growth and survival signaling. Among that being Bruton's tyrosine kinase, of course, being the target of ibrutinib. So this was, from the get-go, an actionable mutation that we sought to identify. And so this, was rapidly translated into clinical trials that have looked at BTK inhibitors, ibrutinib as monotherapy, ibrutinib in combination with rituximab, and now we're also seeing new BTK inhibitors like zanubrutinib and acalabrutinib also being investigated in Waldenström's.